Okay. So, question that was kind of asked was specifically about kind of the different types of contractions. We're going to go over those really quick. Pull up a little document for us to look at. Okay. So our different types of contraction. We have our concentric, eccentric. These ones talking about. And then there's their isomet. Do they do isometric, isotonic?
There was 3 questions on that kind of stuff.
Of like including all these.
I mean, I can't see your screen. I'm sorry.
Oh, sorry, my bad.
But I know there was a concentric, eccentric question. There was a isometric question. But then I was talking more about like the steps of muscle contraction, like ACH opens up the ligand channel. Like it was more stuff like that, I guess.
Yeah. Let's go over these really quick, and we'll just do a quick review of the step-by-step stuff, because it's super, super important that we look at the step-by-step stuff, because that all of that with skeletal muscle, with smooth muscle, with cardiac muscle, all of that stuff is going to come back around. So all of this material from exam 1, don't just forget about it, especially when you get your grade back. If you can review the exam, make sure that you do, because you need to see kind of what information maybe you didn't understand, because a lot of the information is going to come back around. And if you didn't get it on the first exam, the second exam and the third exam and the final, everything, it's going to come back. So we just want to make sure, that you understand it the whole time.
Yes.
Let's say of skeletal muscle, smooth muscle, and then cardiac. Okay. So really quick, what things to remember about this, about all of these words. First and foremost, ISO. What does ISO mean? We've talked about ISO a handful of times. What does ISO mean? It means same. Right? So literally what these words are saying, metric system is a unit of measurement. So same measurement, or in other words, same length. So that means the length of our muscles don't change. So they're not contracting, they're not shortening, and they're not lengthening, right? So if we stop and we think, what's an example of an isometric style of muscle contraction? It could be something, for example, like pushing against a wall, where we're not moving, the wall's not moving, or even it could be like if you've ever heard of the exercise isometric holds, where you take a weight, you get it up to a certain spot, and you hold right there. So what it means is that we're exerting a force. So we are creating a force, but that force is matching whatever, or is matched by whatever we are acting on. So isometric, the length is not changing. Okay. It's like, go back to the trying to push the wall example. You can push really, really soft, or you can push really, really, really hard. No matter how hard you push, you're not going to move, and the wall's not going to move, or at least it shouldn't. Those are going to be our isometric type movements. Concentric, this is when the muscle is contracting and the muscle is shortening. Contracting. The muscle is shortening. So this is a lot of our exercise, like the first part of our exercise, right? For example, like a dumbbell curl. The part where we're coming up to bring the dumbbell up, that's our concentric muscle contraction. Anytime that the specific muscle that we're activating or working shortens. Eccentric, the muscle is contracting and the muscle is lengthening. So dumbbell curl up, dumbbell curl down. When we go down, right, we're still controlling the weight. So we're still exerting a force, the muscle's activating, but we're allowing gravity to win. So as we control that movement all the way down, our bicep muscle gets longer and longer and longer, and we have control all the way down. Isotonic means same force. Let me double check. I want to say iso. Isotonic, we say we're using the same force or constant force. Really, a lot of different exercises can be isotonic. Walking, running, hiking, swimming, skiing, dancing, because the tone overall is remaining the same. The amount of force, like walking, if we're just walking at the same pace or running at the same pace, the amount of force that we're generating is staying consistent. Isokinetic, meaning same speed, so same force. So performing actions isokinetic refers to same speed or velocity. This is only done with very specific machines for very specific rehab. Did he ever talk about isokinetic stuff in class?
It's just mentioned in the slides like once.
Yeah, so it's very, very specific. This is the only kind of question you need to ask about. So ISO. They're very, very fancy, very, very expensive machines. So this is what they look like. And what it's designed to do is to always go the exact same speed. So no matter how much force you apply to, in this case, to the pad, going like backwards, going down for this person, the pad is always going to resist at enough force for it to move at the exact same speed all the way through. So literally the only thing you need to know about isokinetic is that it requires the specific machines, and it's specifically done for rehab. A lot of times with elderly people, it's done with rehab. So, and it's also it's not very common. We don't talk about it a lot because it is so freakishly expensive. The machines are. And so there's no real reason to do it. Okay. Any questions about those muscle those types of muscle movements.
All good there.
Okay. Quick, let's go over and review our step-by-step of our all of our movements. I'm not going to write them out, but I'm going to draw them because it's a little bit easier. I'm going to go all the way. We'll start all the way from the beginning. We should probably do step-by-step of action potential as well. Joseph, you mentioned he had questions on that, right, as well?
Yeah, on both.
Okay.
He actually had a table for action potential, so.
Okay. Was it kind of like a step-by-step table? Something, or what was?
Yeah,
roughly?
It was like between this and this, what would happen on the table, basically.
Okay. So, action potential. Easiest way to remember action potential is to draw our little chart. This little dashed line is our threshold. This is our isoelectric point. So this is our resting membrane potential. And we start here, we're at our resting membrane potential. What is responsible for keeping us at resting membrane potential? What is the specific protein that does that?
ATPase.
Yeah, our sodium potassium ATPase. It's important to remember those AKAs. They can say this is sodium potassium pump, sodium potassium ATPase. Sometimes they just call it sodium potassium. I started says sodium potassium pump or sodium potassium ATPase. There's another name that includes like ATPase or something, but just be specific. Make sure you remember those AKAs. Next step, we get some kind of stimulus, right? Some kind of stimulus that causes an influx of sodium through what kind of channels?
Voltage gated.
Not quite yet voltage gated channels. Remember, voltage gated channels are only going to occur as soon as we hit threshold. So before voltage gated, what are the types of channels that allow for sodium to pass through?
Just the sodium
Ligand.
channels? Oh, ligand channel.
We call them leaky
channel.
channels. Yeah, right?
The leak channels, okay.
The leaky channels. These are the ones that they don't require ATP or anything else going on, but they're caused by any other kind of stimulus. They allow sodium to diffuse. Remember, diffuse means just going straight through, not needing ATP. They diffuse through the sodium leaky channels to allow us to get up to threshold. And this is from those stimulus that we get. And so, for example, we have a small stimulus that happens here. We go back down to resting membrane potential. We get another stimulus, and we get another stimulus, and all of a sudden we get more stimulus, and we hit threshold. Now, the second we hit threshold, what happens at threshold?
Depolarization.
Depolarization, right? Boom. A little too steep, but just like that, we get depolarization. What causes the depolarization? What's happening right here that causes depolarization to occur?
The inside of the cell being more positive.
That's the process, right, that takes place. The way that happens is that we get an influx of sodium. So at this point at threshold, our voltage gated sodium channel activation gate Remember when we talked about the different gates of sodium? Like how here's the, let me see this. The sodium ion, kind of these two parts. This is what the sodium ion looks like. We have our, this was our, we have our activation gate and our inactivation gate. Does this look familiar?
Yes.
So as soon as we hit this point here in the threshold, our voltage gated sodium channel activation gate opens. And we get, boom, depolarization. As we continue to go up, right, we hit the 0 mark. And somewhere between here and here, and the very, very tippy top, we have our voltage gated potassium channels start to open. Okay, I believe, let me actually look up exactly at what voltage, let me pull this over so you guys can see it. At what voltage do voltage gated potassium channels open? I don't know if you got this specific, but voltage gated potassium channels start to open somewhere in here, somewhere between threshold and the very, very top of the very end of depolarization. They start to open somewhere along here. We typically say at the very, very top. This gets all the way up here to the top, right here at this point. Oh, I just deleted. At the very top, the very crest, right here is when they finish opening. So we said the voltage gated potassium channels open at this point. And once our voltage gated potassium channels are open, potassium starts to rush out of the cell. And what happens then? We get repolarization with hyperpolarization before it finally goes back up to resting membrane potential. So right there. So it's important also to remember our voltage gated sodium channel activation gate open down here. So same thing up here. So up here at the top, we get the potassium gate finally opening, but we also get the voltage gated sodium channel inactivation gate closes. It closes and inactivates that channel, so that way sodium can't keep passing through. So we have both of these things occur up there towards the top. Then we get repolarization. The sodium goes, potassium is going out of the cell, out of the cell, out of the cell, keeps coming out of the cell all the way to we get down here towards the bottom. And finally, our voltage gated potassium channels close. And then finally we get brought back up to resting membrane potential. What helps us get up to resting membrane potential and stay at resting membrane potential?
Sodium potassium pump.
Yeah, our sodium potassium ATPase, the sodium potassium pump. Make sure you know this step by step. This is a really good thing. Each of these steps, kind of what happens before this, what happens after this, and each of these steps Very likely test type questions. Like right here, the voltage gated sodium at threshold, the voltage gated sodium channels, voltage gated sodium channel activation gate will open. What is the next step, or what is the next thing that occurs? Next thing that occurs could be the first best answer is going to be depolarization. The second best answer would probably be something about the voltage gated potassium channels opening. Okay. Any questions about our action potential stuff?
No.
Okay. Remember, best way to remember this and understand this is to draw it several times. Just adding in here as well. Remember that between these 2 points, right here, we have our absolute refractory period. And then after that, we have our relative refractory period. There's usually a question or two. What are you? Go away. There's usually a question or two about those, kind of what they are. Absolute refractory period, meaning that no other action potential can occur. Relative refractory period being that it's more difficult for an action potential to occur during that time because we're hyperpolarized. Okay. Next one. Muscles. Skeletal muscles specifically. Start all the way over here in the corner. The action potentials, right, are occurring in the neurons. Those neurons continue to travel. A neuron is shaped like this. We have our soma, which is the body. These dendrites coming out, which receive signal. Then an axon that comes out like this, and then it actually breaks off into axon terminals. Okay, that is our nerve cell. We're going to focus, actually, keep all that. We're going to focus right in to right here, our axon terminal. That action potential that we described in those gates and stuff opening and closing happened right here. That signal travels all the way down here, continues down, and we're going to see that signal coming down right in here. So all of our voltage gated sodium channels and potassium channels are here on the sides, and they're opening and closing and doing their thing and letting this signal come on down through. Within the axon terminal, we have special channels. These are our voltage gated calcium channels, right? What's the purpose of our voltage gated potassium channels? What function do they play? What's their role?
The exocytose in
Exocytose what? What are they helping to exocytose?
The calcium?
Very close. So the calcium comes in, so we are voltage gated calcium channels.
Or is
and
it
the
the
signal comes down.
ACH?
Yeah, the ACH. The voltage gated calcium channels open and they let calcium come on in. And the calcium comes in and interacts with these bubbles called vesicles. The vesicles, inside of the vesicles, holds acetylcholine, or ACH. The calcium comes and it binds to the outside of those vesicles, and that gives it a message that, like, hey, it's time to go to work. And these are going to come and they're going to exocytose. So the membrane of the vesicle merges with the membrane of the axon terminal, and which releases, so it looks a little bit like this. It merges kind of like that and releases our acetylcholine. it releases it specifically into this space known as the synaptic cleft. Within this whole big thing, which is called our neuromuscular junction. When it gets released into this space, across that cleft, there are a bunch of channels waiting to accept it. These channels are our ligand-gated acetylcholine channels. Okay, so the ligand that is gating these channels is going to be acetylcholine. When acetylcholine comes and binds, specifically these are ligand-gated acetylcholine cation channels. They call them cation channels because they let sodium come in, they let potassium flow out. So because they're letting multiple cations in and out and come through, as well as a little bit of calcium to come in, we call them cation channels because they let both sodium and potassium cross in between them. Now, all of that sodium that's rushing in, coming through, makes all of this area more positive. And as the sodium comes in, it kind of bleeds over off to the side and off to the side, just like before in our nerve cell. We have our voltage-gated sodium channels and voltage-gated potassium channels that open and close exactly the same, just like it was happening with our action potential. So literally, once we get to this point here, to travel along the membrane, also known as the sarcolemma, that's the plasma membrane of the muscle cell, we can literally draw the exact same graph that we had earlier, right? There's threshold, our resting membrane potential, and literally we get the stimulus that comes through, bam, we hit that, we come back in, just like that. It looks exactly the same. Only difference is that with a nerve cell, our resting membrane potential is 60 to 70 millivolts, and our skeletal muscle is negative 90 millivolts. That's really the only differentiating factor between the two. Otherwise, this entire process right here is exactly the same as in the nerve cell. This exact same thing, same channels, same everything open to transfer and pass along the action potential along the sarcolemma. So it continues along the sarcolemma until finally it hits a tube that carries it down into the cell. What's that tube called?
The T-tubule.
Yeah, the T-tubule. On either side of the T-tubule, we have these funky things. What are on either side of the T-tubule?
The cisternae.
Yeah, the terminal cisternae. The terminal cisternae are just the end portions of what is known as the sarcoplasmic reticulum. Kind of like in cells and tissues that we talked about, try one. Sarcoplasmic reticulum has the same job as the endoplasmic reticulum. The endoplasmic reticulum, remember, in the cell, its job is to store calcium and release calcium when it's needed. The sarcoplasmic reticulum does the same thing, It stores calcium, holds onto it within the muscle cells, holding on to it until it is needed. Sweet. So our action potential now has transferred across the muscle. And it's traveled across the sarcoplasmic reticulum, and now it's gone down the T-tubule. In the T-tubule, we're going to zoom in to this space right here, make this a little bit bigger, right? So within the T-tubule, on either side, we have these sensors, these voltage sensors, right? Can anybody remember what these voltage sensors are called?
DHP.
Yeah, what's the long name of DHP? What's that abbreviation for?
Dihydropyridine.
Yeah, dihydro. It's either dihydropyridine or dihydropyrimidine. Dihydropyridine, I think, is correct. DHP, these are voltage gated sensors. So our voltage change comes down. Remember, these are all the same. This is all the way through. These voltage gated sodium and potassium channels follow all the way through here. When it comes through, the DHP senses like, hey, the voltage changes. Because remember, we got these channels all the way down on either side coming through. So this lets sodium in, gets more positive. The DHP senses like, yo, it's more positive. And it helps activate another receptor that it's attached to. This is a mechanoreceptor. These are sarcoplasmic reticulum. Who remembers what the mechanical receptor is called?
The rye or something rye one.
It's ryanidine. I just
Ryanidine.
call it, yeah, the ryanidine. I just call them the Ryan. That's how I remember it, because of like ryanidine or Ryan or something. But if you remember that they're the Ryan receptors, to call them Ryan. That's what I call them. As soon as the DHP gets active, this mechanism is gonna, the ryanidine receptors are gonna activate, and that pulls this off and away. Pulls it tight like that. Let me see if I can. So we end up opening a hole in the sarcoplasmic reticulum. When that hole opens, let's go dark blue. When that new hole finally opens, a whole bunch of calcium that's stored inside gets to escape and go out and go all over the place. Now this is where it gets super fun. This calcium, what does this calcium do? Where does this calcium go? And that...
To the
Go ahead.
troponin.
Yeah, right, because we have our actin and our myosin filaments. So we have our actin. And it looks like this. And then, of course, our actin has troponin on it, which is 3 parts. And then we have our nice myosin fibers down here as well with our myosin heads. Like that. On each of these actin, we have this binding site on there. What's the name of that binding site?
Is it just like the myosin binding site? Oh.
Yeah, good. That's what I wanted to do. I wanted to make sure you guys got that correct. the myosin binding site, because the myosin is going to bind onto that site located on the actin. Good. So all these little myosin binding sites are there. And then we have big bad tropomyosin doing its thing, blocking all of these binding sites. Just like we said, we have our calcium. Our calcium comes down, binds onto our troponin. Specifically, what part of troponin does it bind onto? There's 3 parts of troponin, right? Does anyone remember the 3 parts of troponin?
Is one part of it titin? Titin?
That's not part of the troponin. Titin is another protein that actually helps with the elasticity of the muscle tissue. So we have, kind of zooming back out a little bit, we have our sarcomere right here, and we have our actin thin filaments, right? And then we have our myosin thick filaments, just like this. And this isn't perfect. Titin.
Oh, sorry.
You're good. Come on. Titin is a little protein that binds to the myosin and then binds it to the Z disc. Like this blue guy. This is titin, aka connectin, which helps with the elasticity of muscle. It helps muscle return to its normal shape after contraction. So that's titin right there. Okay. These guys are titin, aka connectin. We're going to be talking about troponin. So troponin is a little bit different. Troponin has 3 components. Troponin C for calcium.
Oh, yeah, I was thinking, yeah.
Troponin I for actin. And then troponin T for the tropomyosin. And what it's literally telling us, the troponin T, troponin C, troponin I, it's telling us what that type of troponin binds to, what it holds on to. Troponin C is able to bind to calcium, troponin I binds to actin, and troponin T binds to the tropomyosin. So this calcium released from the sarcoplasmic reticulum comes all the way down, binds onto our troponin C, and that causes, kind of like, remember when we talked about hemoglobin in Biochem 1, and when the first oxygen would bind to hemoglobin, you'd get what's called a conformational change, like the shape of the whole structure would change to allow for more oxygen to bind. You guys remember that in Biochem 1 a little bit?
Yes.
Same thing happens here. Calcium comes and it binds onto that troponin C, and it causes a change in that whole molecule so that it changes shape and moves. And in the process of moving, it literally pulls and rips off the tropomyosin off of those binding sites. So then all of a sudden, these binding sites are free and clear to allow for binding. And these myosin heads are down here just ready. As soon as that tropomyosin moves off, these myosin heads snap super quick and bind straight onto that myosin binding site. So snaps and binds right there, and then it pulls. So it's laying down, it's resting, it's waiting. The actin, the binding site, myosin binding site on the actin opens up, it snaps onto it and pulls. So it's resting, that binding site opens up, snap, pull. That pull is called a power stroke. Okay, and that is how our muscles contract. Is thanks to that pulling, that power stroke of the myosin. That's not all, though. We have one last step because we need to get the myosin head off of the actin and reset it so that it can do that again to let our muscles keep moving and contracting. And the way that that happens, I'm going to take another box right here. So we have our actin, we have our myosin binding site. Our myosin is bound right onto it. And the way that we release this is with our energy source, ATP. ATP is going to come in and bind onto the myosin head where it has a little spot just waiting for it. As soon as it binds to that myosin head, it's going to break down, it's going to dephosphorylate into ADP and a inorganic phosphate. And our myosin head is going to release, and it's going to cock back, kind of recock, kind of think like a gun, or like the hammer on a gun. It cocks back, it resets back to its original starting position, so that way it can bind to the next available myosin binding site on the actin. Pull again, ATP comes and binds, it releases, and then it resets, snaps back, binds to an actin, pulls, and then a new ATP comes, binds to it, gonna let go and reset, binds to a new actin, pulls, and so on until the muscle contraction that we have is over. And the way that all of this process stops is that we have to stop it all the way up at the beginning, all the way up in here, so the signal stops being transmitted. And so we release an enzyme called acetylcholine esterase. Acetylcholine esterase comes in, it breaks down all the acetylcholine in the cleft, helps it get reabsorbed back to be recycled and reused to stop the whole signal from happening. The ryanodine channels, these guys will close because the DHP are no longer receiving a signal. And then along the sarcoplasmic reticulum, we have pumps. These are calcium ATPase pumps, which grabs all of the calcium and pumps it back into the sarcoplasmic reticulum and stores it in a protein called calsequestrin. And that's our whole muscle process. That was a lot. So any questions about the step-by-step muscle process of this process of contraction of a muscle cell?
No.
Okay. Again, kind of like what I just did, be a good idea to be able to, kind of draw this out, practice this, because understanding this process forwards and backwards is the easiest way to understand is the easiest way to answer test questions when it comes to the actual test. Because then okay, the signal enters into the T-tubule, what's the next step that's going to occur? DHP is activated, which activates the ryanodine receptors, which then causes calcium release from the sarcoplasmic reticulum, and then that calcium goes and does what it does. Or they can say, before the acetylcholine is released into the synaptic cleft, what voltage gated channels are responsible for opening to allow exocytosis? And those would be those calcium gated, or those voltage gated calcium channels way back up in the axon terminal. And so on and so forth. All that good stuff. I'd probably spend more of my attention. Not as much here, this stuff. More from DHP on. Make sure you really understand the makeup of actin and myosin and kind of how this whole thing happens, how the calcium comes and binds, pulls off the tropomyosin, and the myosin head binds in power strokes and then releases once ATP binds to it. I'd more focus on those guys, and then how the whole thing ends with acetylcholinesterase and the calcium ATPase bringing in all of the calcium. Because once we clean up all of the calcium, our troponin C is no longer has calcium in it. So then the tropomyosin is going to go back to covering up all of those myosin binding sites. Cool beans. Joseph, don't want to put you on the spot again, but do you know, did Dr. Tate get into on the exam a lot of the, like, during contraction, what happens to all of the different bands and zones of the sarcomere?
The very first question was about the I band and if it shortens or not.
Okay. Make sure y'all review
Other
those.
than that, no.
Okay. I just reviewed those. My guess is that he probably either has a question bank, so multiple tests that are different, or he has different tests for the blended. So don't go off of, what people from the in-person say versus the blended. They're two separate programs. My guess is that they're going to be slightly different exams. So, okay.
It was also one where he drew out the sarcomere and like it didn't say it was the I band, but he like bracketed it in and was like, does this shorten or not?
Okay, yeah. I guess just as a quick review, here's our sarcomere. 1, 2, 3, 4. 2, 3, 4. 1, 2, 3, just like this. Little labeling. What are these lines called right here? The up and down black ones.
Z disc.
discs.
Yeah, or Z disc. What about the thin red lines? What are these called?
Actin.
Yep. What about the thick lines?
Myosin.
Good. All this whole zone that has all of the actin in it, what's that called?
I band.
Yeah.
Good. The I band. Because remember when we think about troponin, it's troponin C, T, and I. I stands for actin. What if we just look at this area that only contains myosin?
The H zone.
The A band.
Good.
Oh.
H zone, A band, they're pretty interchangeable. Don't worry too much about that. What about if we include the whole thing of myosin, including some of the actin?
That's the A band.
That's our A band.
Yeah.
If we can draw it like this, think about what happens if our sarcomere contracts. So if this contracts, these come in. What happens to our H zone or H band?
Disappears pretty much.
Yeah, our H is going to get smaller, right? At the same time, this is contracting, right? Because this is all moving inward like this. What's actually happening to our I band?
It's shortening.
Our I band is also shortening. So when the H zone or H band shortens, so does our I band. What happens to the A band? What does the A band do?
It stays the same, right?
Yeah, the A band stays the same because the A band doesn't care If the actin of the myosin is changing, it's just the total length of the myosin, and the myosin itself is not moving. What's the name of the protein that connects the myosin to the Z disc?
Oh, this is the titin.
Yeah, titin, titin, also known as connectin. Any of those names? However you want to pronounce it, say it, whatever. It's fine. I think there's also technically like an M line or something.
Yeah.
Yeah. Oh, you never get asked with the M line though. How do we, looking at this picture, how do we measure the sarcomere from where to where?
Z to Z.
Z to Z, good. And just another note, I don't know if he asked this question or not, but remember that the sarcomere is the functional unit of the muscle cell. So if you talk about functional unit, it's the sarcomere. What is a motor unit?
Oh, it's
The
is it
neuron and all the muscle fibers.
yeah, I was gonna say all of everything.
Yeah, an alpha motor neuron and all of the muscles that it innervates. Okay. Another one. Let me pull up.
Can we go
It's
over
been a
like
while.
the knee-jerk reaction too?
Knee jerk?
Yeah.
Yeah, we can jump to that. One sec, let me. Where's my. Real quick, I'm going to go over the smooth muscle contraction. Smooth muscle is a little bit different. And I like this picture right here. Smooth muscle isn't super complicated, but I just want to point out a couple things. We're going to do smooth muscle real quick, and then we'll. So I don't think do you guys actually get to cardiac, like the whole contraction process of cardiac? Did you guys get to that?
No, I think it's just mainly knowing the differences between the
Yeah.
muscles.
Yeah, just like, okay. So smooth muscle, biggest tip here first like to start out, remember where smooth muscle is found. Kind of think about what its job is, what its function is. The easiest one to remember that I think about is the gut, right? So we think about the intestines and all that. What's the job of the intestines? Its job is literally to squeeze and push and move food along. So that way, you know, kind of squeeze and squish it up and mash it up to allow us to absorb nutrients and just keep it moving all the way through our entire intestinal system in order to excrete it out. So the way that it starts, this whole smooth muscle process is based on calcium. Okay, we have these calcium channels right here, and these are mechanical calcium channels. What does that mean? It means that when they get that tissue gets pressed or stretched, these channels activate. Okay? And so what presses or stretches the intestines? When they have food and stuff going through them. So as soon as they get pressed or stretched, you get an influx of calcium ions coming in from the ECF. ECF stands for extracellular fluid. So just the fluid that's outside of the cells. It's full of calcium, especially in these areas. So calcium comes in. And then when this calcium comes in, it creates a reaction and it causes the sarcoplasmic reticulum to activate and release even more calcium. So calcium comes in because of the movement, which ignites the sarcoplasmic reticulum to release even more calcium. That calcium is going to go and interact with a specific protein called calmodulin. That calmodulin gets activated when calcium binds to it. Then we have a kinase. This is the specific name of the kinase. I always remember it as the Martin Luther King kinase. Yeah, MLCK. Called MLCK. So it's what does it actually stand for? It was not something.
You had it right there, myosin light chain.
Myosin light chain kinase. Yeah, it's not. Yeah. Wherever I had it. Yeah. Myosin light chain. It's myosin light chain kinase. Okay, so the myosin light chain kinase gets activated by that calmodulin. Oh yeah, right there. Activates the myosin light chain kinase enzymes, and that catalyzes the transfer of phosphate to the myosin heads. The breakdown of ATP activates the myosin heads, and those form those cross bridges with actin and thin filament, and then the shortening occurs. I feel like smooth muscle is a little more straightforward than skeletal muscle. The really big things you need to remember the names: calmodulin, the MLCK, and then those are the big ones to remember. Remember those two guys when it comes to smooth muscle stuff. Another thing to remember about smooth muscle is the contraction of smooth muscle really, really fast or really, really slow?
There was something
Slow.
inarticulate. Oh, slow.
It's really slow, and it's also sustained, meaning that it's super fatigue resistant. So the contraction, it squeezes really, really slow and for a long time. So it'll squeeze, and hold for long periods of time.
I feel like he covered smooth muscle very heavily on the test too.
Did he?
Was there anything? Or sorry, go ahead.
Was it mostly this kind of process, or was it other kind of?
Yeah, like myosin light chain kinase was on there. Calmodulin. There's like at least 4 or 5 questions about it, I think.
Unarticulate. It went into like the smooth muscle in regards to the norepinephrine, and then there was like 2 different I guess reactions that could happen. One was inhibitory. I wasn't sure if I really needed to memorize. I forget what they were. They were like alpha, beta, something. I'm not sure.
Alpha gamma coactivation.
I remember that, but I think it was.
There's
It wasn't
this.
that. It
So
wasn't that.
this is part of it, to knowing that acetylcholine activates the gut system and norepinephrine inhibits it. There's that. Let me see. Talking about like this stuff with G protein coupled receptor stuff getting in there. which is
It's
I
I,
remember
I remember
doing
it now. It says alpha 1 adrenergic receptors and then
Oh,
the beta
adrenergic.
2. Yeah.
I wouldn't worry too much about that. That's so what that's referring to, there's different types of receptors. There's alpha receptors and beta receptors. So like there's a couple key words that you'll see, like you'll see it's like the naming of different types of receptors. So you'll have alpha, you'll have beta, you'll have adrenergic, you'll have muscarinic. Gosh, how do we spell it? Alpha, beta, adrenergic, muscarinic, cholinergic, all these kind of words. Don't worry too much about that right now. Occasionally you'll see things like that. These names pop up. Whenever they do pop up, you can take another second to kind of look at them. But you'll talk a lot more about alpha receptors, beta receptors, and kind of their jobs and what they do and the different types in more of like the pathology and pharmacology courses.
Okay.
That's where you talk a little bit more about those.
Okay.
So I would remember like yeah, whenever you see these if you see these pop up again in Phys 1 class, they're not going to give you like specific questions about like these names, but if you see them pop up here and there, take a second to kind of look them up a little bit and kind of see kind of what is going on and start to build your own base knowledge of these. So that way in the future, you have a little bit of a foundation to go off of. Okay. Any questions at this point about smooth muscle and all that stuff? Make sure you know the differences between smooth cardiac and skeletal muscle. So like smooth muscle only has one nucleus. Cardiac muscle can have one or two nuclei. Muscle tissue has a ton of nuclei. Muscle contraction, skeletal muscle is voluntary. Cardiac and smooth muscle is involuntary. All those kind of comparison things. Teachers love those for tests. And you can see here kind of the cycling of the cross bridges. So the cycling of the contractions is a lot longer. It lasts for much, much longer time. He potentially could ask about this. Acetylcholine versus norepinephrine. Easiest way to remember that is typically like, when do you feel like you need to go to the bathroom? Typically it's when you breathe, relax, after you've just eaten and your body's digesting, those kind of things. Acetylcholine is a lot of acetylcholine is more of our sympathetic type system, the autonomic nervous system. And then the norepinephrine, aka noradrenaline. Adrenaline, if you think about it, if you're running from a bear, the last thing that you're thinking about when you're running from a bear is, oh, I think I have to go to the bathroom. So yeah. Okay. You had said knee-jerk reflex, JC, right?
Yeah.
Okay. First things first, let's talk about this thing called a reflex arc. The reflex arc is kind of the main steps that we're looking at of how this whole process happens. Right, so first things first, we have we're going to begin with a stimulus happens, right? And we're going to end with some type of reaction. Neither of these are part of the reflex arc. They're just how it begins. Okay? The reflex arc starts with our receptor, main receptor that we're talking about. If we're talking about inside of the muscle belly, what's the name of the receptor that's inside of the muscle belly that measures the speed of our contraction and the length of the muscle? Yeah, these are going to be our spindles. Now the spindles are going to be connected into the spinal cord. And where does sensation enter the spinal cord?
The dorsal sensory side.
Yeah, so we have our dorsal horn. Is the dorsal horn white matter or gray matter?
It's gray, right?
Yeah, it's gray matter. So easy way to remember that is it's reversed in the spinal cord and the brain. Inside of the spinal cord is gray matter, outside is white matter. And then in the brain, outside is gray matter, inside is white matter. So they're flipped between the brain and the spinal cord. So it goes into the dorsal horn. What's the name of the neuron that lets it get there? What type of neuron comes from the muscle spindle? What type of neuron?
Alpha motor.
So we think about it,
Or
right?
type
It's going into the sensory portion of the spinal cord. Alpha motor neurons come from what part of the spinal cord?
Oh,
The ventral.
ventral part.
Yeah, ventral, the anterior, right? So the type of neuron that's going into the dorsal horn
Sensory.
is going to be our type 1A sensory neuron. Okay? And we name these. There's type 1A, type 1B, technically a type 2. We don't go super deep into those. Don't worry about it all that much. JC, did he ask about spindle versus or chain versus bag fibers?
I don't think so.
Okay, I won't worry about that then. Okay, so the spindle receives a signal. It sends it along the sensory neuron, and it goes into the spinal cord, and it's going to synapse with our alpha motor neuron, which goes and it brings it to the muscle. The alpha motor neuron brings it to the muscle, whatever muscle we need to have do whatever. And then we finally get that reaction, typically some type of contraction. So the reflex arc starts at the receptor and ends at the target muscle. This is our alpha motor neuron. Okay. This is the rough overview of the reflex arc. Let's get into specifically the knee jerk reflex. So the knee jerk reflex is testing the L4 nerve root. You don't need to know that right now, just prepping you for the future. L4 nerve root. So in the knee jerk reflex, we take our little reflex hammer. Ooh. So pretty. And we hit the knee. Here's our beautiful knee, the little foot. Whoop, whoop. We hit the knee right there. Bam. There's a patella just below the patella on the patellar tendon. As soon as we hit it with the hammer, what's actually happening when we hit it with the hammer?
Stretching the muscle spindle.
Yeah, we stretch the muscle itself, right?
Yeah.
As when we hit it with the muscle with the hammer, essentially what that's doing is kind of like, I don't have anything good, put a good example here, but if you push something is affixed at both ends and you push one part of it in, it's going to tighten and increase tension, and change the length and change the, it's going to quickly change the length and the amount of force, whatever, that's on the rest of that that muscle that's connected from both ends. As soon as we do that, our muscle spindle fiber is sitting deep inside of our quadriceps muscle, right? And remind me, is our muscle spindles intra or extrafusal fibers?
Extrafusal.
Extra.
They are intrafusal. Muscle spindles are intrafusal because,
Oh, yeah, makes sense.
yeah, if remember we talk about fusiform. Fusiform is kind of a shape. It's kind of like that diamondy sort of shape, kind of like sideways, sideways like human eye or whatever. It kind of looks like that. That's what fusiform means, and that's the rough shape of the muscle spindle. And overall, kind of the rough shape of most of our muscles, right? Because we have an attachment site right here, and like the whole muscle belly, and then an attachment site up top, right? More or less, our muscles are kind of fusiform shaped. So intrafusal, meaning that they're inside of the muscle tissue. They're deep inside, because they're deep inside of the muscle tissue, doing all the monitoring, all the checking, making sure things are doing what they're supposed to be doing. The extrafusal. Go ahead.
Sorry, the intrafusal is just the bag and the chain fiber, right? Like, in essence.
Mm-hmm. Yeah.
Okay.
The bag and the chain
I just want to
fibers.
make sure.
Yeah. Intrafusal are the bag and the chain fibers. Extrafusal is all of the muscle tissue around it.
That
and
makes
what
sense.
connects to
Okay.
it. Yeah. So just like we said before, we have our muscle spindle. The muscle spindle senses that there's been a change, so it changes the way that it communicates. Now, if we look at a graph at the muscle spindle, it's not that it's either sending a signal or not. It's always sending a signal. It just depends on how quickly it's sending a signal that the brain translates that into what's going on. So if the muscle is sitting at rest, so like my arm, if my arm is sitting right here, camera don't work. Arm is sitting right here, that's fine. Even if I change my position, if I prop up my wrist like this, and I have it propped up, hold it right there. Even though the length or whatever the muscle is different from each position to position, both of them are at rest. So the muscle spindles are at rest. So the muscle spindles are sending a slow, constant rate. This might be really stupid, but for some reason, the way that I picture this and remember it, if anybody ever watched SpongeBob, there's a part where SpongeBob is with the Flying Dutchman on his boat, and him and Patrick are like backing the boat into a reef, and it's just like SpongeBob saying, "You're good, you're good, you're good," over and over and over again. Does anybody know what I'm talking about?
Yeah, I got you.
Does anybody else know what I'm talking about? Or is that not something?
I mean, I've seen
I didn't have
SpongeBob, so I can
cable.
imagine it.
And then he
Yeah.
just keeps going. He
Same.
backs it through the wall.
Let me, because for some reason, this is what like really sticks in my brain. And it's really helped me it really helped me grasp the concept of of kind of what the muscle spindles are doing. Let's see if my computer, I've got like 8,000 tabs open right now, so it might. So this is kind of the premise where it's like, you're good, you're good, you're good, and then stop. So for some reason, that's kind of the job of the muscle. That's silly, but that's the job of the muscle spindles, is literally just to tell the brain, like, hey, you're good, you're good, you're good, you're good, you're good. And it's constantly sending that signal, just like checking in, just like, hey, we're still good, we're still good, we're still good, until we move, or in this case, the knee-jerk reflex, until that hammer hits that tendon. Technically ligament, if you really want to be specific, but tendon, ligament, science, the debate on that is still big. But as soon as we strike and that gets hit and the length of the muscle changes, you get an increase in signal. Suddenly the muscle spindle is sending a rapid signal, letting the brain know, like, hey, something's changing, something's wrong. And eventually this signal will get to the brain, but the body is built to protect itself in order to prevent injury from happening. So we have these spinal cord reflexes that allows our body to act and to respond before the message goes all the way up to the brain processes it and sends it back down. Okay. And so what happens, we have our type 1A sensory neuron. I'm going to redraw this entire thing, basically. Goes into the dorsal horn of the spinal cord, and it synapses specifically with an alpha motor neuron. That alpha motor neuron comes back to the quadriceps, goes to the extrafusal fibers. This is our alpha motor neuron. Goes to those extrafusal fibers and tells the extrafusal fibers, hey, you guys need to grab on, quickly contract, to make sure that our muscle doesn't stretch too quickly. Because if it stretches too quickly, we could straight up tear it, or it could get ripped apart, or any number of things can happen to it. So we need to make sure that we protect it. So the alpha motor neuron runs back to that part of the quadriceps and tells the quadricep muscles to contract. And that's why when you hit that tendon, your foot just quickly kicks out, because the quadriceps contract. That is, in its entirety, that is the actual spinal cord reflex. Okay, because that is the arc itself. There are extra parts to it that we also need to know. So for example, off of this same sensory nerve, it comes over as well, binds to an interneuron, an inhibitory interneuron, and then binds to another alpha motor neuron that's connected to the hamstrings. Make sure that I don't say something incorrect. So this connects to another alpha motor neuron, and it inhibits, and it's essentially this interneuron right here blocks any other signals from traveling down the hamstring alpha motor neuron and forces this whole muscle to relax. So that way this muscle the quadriceps can contract and the hamstrings are forced to relax. So that's one step that occurs. The next part is we also have another one coming out here that binds to a gamma motor neuron, which goes back to where the muscle spindle is. This is a it's lambda. Frick, what's gamma? Gamma, beta, omega. That gamma? That's lambda. What's the symbol for gamma? Forgot. There it is. It's the Y. These are gamma neurons. They come back. They run to, they specifically go to the extrafusal fibers that are surrounding and that connect to the intrafusal muscle spindles. Okay. So the way that our whole muscle is set up, we have our whole muscle cell right here. Not cell, but the whole muscle, a bunch of those muscle fibers running all the way through, et cetera, and then we have a muscle spindle in here. On either side of the muscle spindle connected to it, we still have muscle fibers connecting to it like this and continuing along its path like that. So this gamma motor neuron comes to these extrafusal fibers that are attached to the intrafusal muscle spindle, and it tells those muscles specifically to contract in order to match all of the rest of the muscle fibers to keep the muscle spindle at the proper tension, at the same, I should stop saying tension, at the same length, keeps the muscle spindle fibers at the same length as all of the rest of the fibers, as the rest of the muscle, in order to continue to send the proper signal up to the brain that everything is okay. Questions about, and that was a lot of information, questions about this whole process?
What about like the alpha gamma coactivation? Like, how does that like work?
Mm-hmm. So literally what alpha gamma coactivation is it's this alpha motor neuron right here. The alpha motor neuron that's running to that muscle, and then the gamma motor neuron running to the same muscle. So all that is what alpha gamma coactivation is saying. It's literally just saying that at the same time as you get that activation of the muscle from the alpha motor neuron, you also get the gamma motor neuron coming in and activating those things to do what I just described, to keep everything the same length. So that's all. That's just putting the name to it, saying that. So this whole process occurs, and they call it alpha gamma coactivation because both of those happen at the same time. Does that make sense? It's just the label that they've given it.
Mm-hmm.
Okay. Any other questions about this whole process? This gets heavily tested on your first lab exam. Make sure if you review your first lecture exam and come back to this stuff, because a lot of this muscle stuff, a ton of this muscle stuff, like alpha gamma coactivation, the muscle spindle, Golgi tendon organ, the different alpha motor neuron, sensory neurons, extrafusal, intrafusal fibers, all of this stuff is tested very heavily on your first lab exam. as well as
What does
stuff like
he? Sorry,
ahead.
what does he mean when he says like recurrent inhibition?
Recurrent inhibition. Let me see. Still talking about the same stuff?
Yeah, because he's having us draw the knee-jerk reaction, but that's one of the things that he wanted on there was the recurrent inhibition, and I'm not understanding, I guess, like what he means by that.
Oh, recurrent or reciprocal inhibition?
Reciprocal's on there too. It's reciprocal, recurrent, and then alpha gamma coactivation was the three things he wanted on there. But I don't know what recurrent means, I guess.
Recurrent inhibition. Oh, that's the Renshaw cell stuff. Let me pull that up. Give me 1 sec while it pulls up.
You might be on mute.
I was, thank
Yeah.
you. Okay, so the Renshaw cells, these are small inhibitory cells located in the anterior horns of the spinal cord. It's kind of an off branch. So see how this little Renshaw cell right here, so that one A fiber comes in, it branches, and then it goes down to that alpha motor neuron. A little separate little extra branch comes off of the alpha motor neuron and binds to a Renshaw cell, which then kind of doubles back onto the alpha motor neuron again, to then, as soon as the first signal is sent, it comes in and inhibits itself. So it inhibits the alpha motor neuron to prevent like repeated signals from occurring. So you guys talked about something called clonus at all?
Yes. The ankle one?
Yeah.
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 So clonus would be a good example where kind of that whole process, whatever, gets damaged. And so let's see if this is so see how it goes and goes and goes and goes and goes and goes and goes. This person is lacking that ability to have that recurrent inhibition. Or as well, you can look at like true cases of tetany. Like this is kind of example. The better example is tetany. Jeez. Sorry, guys. Let me assess figure out what it's doing. So tetany, if you've heard of the disease, the condition tetanus, right? Tetanus is an uncontrolled muscle contraction. So it's like a forced muscle contraction that's way bigger, way stronger than it's supposed to be. Geez, okay, everything on my computer is like freaking out right now. Okay. So it's just, so the way that you would draw that is, so you have all of this coming down in through here. And so I would draw a little branch coming out here, connecting to a Renshaw cell and coming back to inhibit. That's the recurrent inhibition to prevent the tetany, prevent the excessive muscular contraction. Does that make sense?
Thank you. Mm-hmm.
Yeah. Renshaw cells are what does that.
And then the reciprocal is just the motor neuron that inhibits the hamstrings from tightening up
Exactly.
or
Yeah.
contracting. Yeah.
Yeah. That is correct.
So could there be a Renshaw on both of the motor neurons, or just the alpha one that goes to the?
So because that other, like we're when we do these drawings, we're looking at a very selective, little teeny tiny piece of the whole thing. So technically, like if we want to get really technical, like here's our horn, right? Just like this. That sensory neuron comes in. And like I said, like we're looking at the super basic version of this whole process. In the future, when you take like neurology and stuff, you'll learn that there are 10 different layers all in through here, that process, and then like this is all split up into different layers, and like this is called rex lamina, and then it splits like this. There's 10 on each side. So like there is so this is the one comes in, it synapses with that alpha motor neuron that goes to the quads, and then a secondary one that comes off, binds to an inhibitory, which then binds to that other alpha motor neuron. Right there, going through. Technically, you also have, there is a hamstring reflex as well, where that comes in and it binds directly to that alpha motor neuron of the hamstring. And then it has one that comes off that then has an inhibitory interneuron that goes to the quadriceps to turn the quadriceps off. So like it's all super interconnected. We break it down super simple and just look at the one specific side of things. So like, yeah, remember there's more interneurons in the entire human body than there are actual neurons. So like,
Okay, yeah, that makes a lot of sense.
I think the number, it was like 100 times. There's like 100 times more interneurons than there are actual neurons.
Good goalie, man.
Yeah,
Nice.
so it's like if you think like even here, like coming off of this thing as well, because technically what happens is, even though the signal passes directly through, another branch comes off and goes out and goes up to the brain, all the way up to the brain, and it crosses across the spinal cord, goes in different parts of the brain, and the brain, it scatters into like 5 different places within the brain to tell us exactly what's going on for the body to process it and process the sensation, process like the muscle movement process, like the plan of action going forward, like all of this stuff. Like it's neurology gets super, super, super complicated very, very quickly. So we take the baby steps.
That's fair.
Yeah.
I just wanted to, I guess I didn't like sit well, and then now it's sitting well. It's sitting well, very well.
Yeah. Okay, good. Any other questions, concerns, other material that you want to look at? For those of you taking the test tomorrow, how are you feeling?
Feeling good.
Good. Like I said, what I personally would probably do is tomorrow before the exam, I would just take a minute and review these main processes. So like the action potential, the whole process of the muscle contraction, the sarcomere, and then like the knee jerk reflex. Oh, and smooth muscle. I would review kind of those step-by-step processes. And with that, that should get you most of the way there for this first exam. All right, if you guys don't have any other questions, we'll go ahead and end the session here.
Thank you.
Thank you guys so much for showing up. Good luck. If you already took the test, JC, hopefully outcome turns good. And then if you're taking the test tomorrow, best of luck. Make sure when you guys start, like when you're testing, if you had some sort of like testing process in try one, try and keep that the same. Keep the same testing environment if possible. Like if you have specific drinks or specific snacks that you like to have while testing, make sure you have try and the more of a consistent environment you can create, the easier your body's going to have like taking exams and the less stress you're going to feel.
Thank you.
Hey, you guys have a great night. I will see y'all later.
Yep. See you later.
Thank you, Drake. Bye.